There's a really great book that I bought. It's called Goodbye Germ Theory by Dr. William P. Tribing. So I can't, oh yeah, you can see it. It's like, there is video on this one. Goodbye Germ Theory. There it is. That's what it looks like. Okay. Because the germ theory is a lie, and we're going to go into that again. And I've talked about it, before, and I'm going to talk about it again, and I'm going to get down to the nitty gritty on it now because it's just those who have a vested financial interest in vaccination programs are given carte blanche authority to declare a need for prevention of diseases that don't even exist. It is a well-known fact that polio was decreasing very well on its own, as well before the mandated vaccination programs. In Europe, where the vaccine was hardly used, this was also the case. In America, polio increased dramatically in the 1950s after the vaccination program for polio was administered. Shortly afterward, once again, if people began showing up with polio symptoms after they had their shot, they were diagnosed with spinal meningitis, even with paralysis. If they did not have their shot, they were diagnosed with polio. How convenient. This is the AIDS US patent, H1N1 US patent, Ebola US patent, swine flu US patent, BSE US patent, Zika patent, SARS patent, and Corona patent. Okay, there's a list. Why? Because they already know that you're going to get these particles from people with certain symptoms because they're under stressful conditions. When you have a bacterium that is a pleomorphic cell, meaning it can change its shape into something else. Like, I'm sure that you understand that bacteria can form spores. You may have heard that before somewhere else. I mean, if you even have a little bit of microbiology, you might understand that bacteria can form spores under stressful condition. Okay, a stressful condition, they go into sporiform shape. That's nothing that's very hard to understand. Is if you have a little bit of microbiology or a little bit of science training, or you took, Science 101, that's part of the curriculum, okay? But what you may not know is that when a bacterium is under stress and they don't have time for the adaptation into a spore-form shape, they begin to make what's called a phage. And a phage is basically saving the nucleic acid or the DNA fragment or RNA fragment, but it's nucleic acid material in a protein envelope that helps to protect that under extreme stress so that later that can feed the existing colonies, and the memory of what that bacterium, that colony, that body, that human, whatever, animal, plant life, etc., can remain. Then it can feed it back and help the bacteria to live properly in the body, as they are part of your own life. They are part of your blood. They give you life. They help you digest nutrients. They help you make vitamins, okay, of your own terrain. Yeah, that's what that is. Under a stressful situation, this is what can happen. And it's a fast stress, not a slow, long stress. And this is the material that they extract and they say is a virus. And the lie is that it causes disease when it does not. Most of the things that they were doing involved just taking cells out of tissue, not in situ, taking them out, putting them in petri dish, using killed products, and trying to come up with answers that way, which are always going to be easily manipulated by whatever outcome you want. This is where he walks through how a laboratory or how the medical establishment has come to some of these conclusions and how they purport the lie. And I'd like to, in working with this video and working with his dismantling the virus theory, using the measles virus as an example, paper that he wrote, as well as bouncing over to Dr. Robert O. Young's information, and I'll show it to you under a microscope what these things look like and stuff so you can get a grasp, okay? So let's go through that. But what I find, before we do, just wanted to touch back on when I was talking about polio for a second, what polio is and what Zika is and what SARS is and what Ebola, all of these disease pictures are toxicity, right? Toxemia. So commonly composed of either DNA or RNA cores with protein coverings and having no inherent reproductive ability, viruses depend upon the host for replication. They must utilize the nucleic acids of living cells they infect to reproduce their proteins, i.e. trick the host into producing them, which are then assembled into new virus-like cars on the assembly line. Theoretically, this is their only means of surviving and infecting new cells or hosts. So this is their theory. The replicating virus theory. Then it was discovered that when bacteria slowly begin to die, bacteria create tiny, apparent lifeless forms of survival, the so-called spores. It was then suspected that these spores were toxic and that they were the so-called pathogenic poisons. This was then refuted since the spores are rapidly developing into bacteria when their vital resources are being restored. When scientists in the laboratory observed that the weak, highly inbred bacteria perished very quickly while turning into much smaller structures than the spores, it was first believed that the bacteria were being killed by the alleged pathogenic poisons called viruses, and that the viruses were thereby replicating. And then there's a picture, a micrograph under dark field microscopy showing red blood cells and the evolution of bacterial phages and bacterial spores, which are these little white spots from red blood cell biological transformation. So then there was the invention of bacterial viruses. Due to the belief that these, at the time of their discovery, still invisible as structures, were killing the bacteria, they were called phages or bacteriophages, which meant eaters of bacteria. Only later it was determined that merely highly inbred and therefore almost non-viable bacteria can be made to turn into phages or bacteria which are being destroyed so fast that they do not have time to form spores. The introduction of the electron microscope led to the discovery of the structures resulting from the biological transformation or pleomorphism of bacteria when these were suddenly dying or when the metabolism of the highly inbred germs was overwhelmed by processes triggered by the adding of phages. It was also discovered that there are hundreds of types of different looking phages. The discovery of phages, or so-called bacterial viruses, reinforced the wrong assumption and the belief that there were human and animal viruses that looked the same and had the same structure. This is and cannot be the case for several different reasons. After introducing chemical examination techniques in biology, it was discovered that there are thousands of types of phages and that phages of one type always have the same structure. They consist of a particular molecule made of nucleic acid, which is covered in a shell of proteins of a given number and composition. It was only later discovered that merely the bacteria, which had been highly inbred in the test tube, could turn into phages themselves by contact with phages, but this never applied to natural bacteria or bacteria which had just been isolated from the natural environment. In this process, it was discovered that these bacterial viruses actually serve to provide other bacteria with important molecules and proteins, and that the bacteria themselves emerged from such structures. Before it could be established that the bacterial viruses cannot kill natural bacteria, but are instead helping them to live, and that bacteria themselves emerge from such structures, these phages were already used as models for the alleged human and animal viruses. It was assumed that the human and animal viruses looked like the phages were allegedly killing cells and thereby causing diseases, while at the same time producing new disease poisons and in this way transmitting the disease. To date, many new or apparently new diseases have been attributed to viruses if their origin is unknown or not acknowledged. This reflex found an apparent confirmation in the discovery of the bacterial viruses. It's important to note that the theories of fight and infection were accepted and highly praised by the majority of the specialists only if and when the countries or regions where they had lived were also suffering from war and adversity. In times of peace, other concepts dominated the world of science. That's also very interesting. It is very important to note that the theory of infection, starting from Germany, has only been globalized through the Third Reich, when the Jewish researchers, most of which had opposed and refuted the politically exploited theories of infection, were removed from their positions. The control experiments show that the phages do not appear if bacteria do not change, or if bacteria randomly start decomposing due to extrinsic sudden annihilation without forming phages. There's pictures now where you see this shift, or the pleomorphic shift, under a micrograph. So this is using a phase contrast microscopy into rod bacteria, cell wall deficient bacteria, Y forming yeast, and then bacteriophages. So they go from one to the other to the other. So then they spin the liquid down and they in a centrifuge, and they take out and they measure the mass. So this is, so why I'm telling you this is because this is how in the lab they're doing this stuff. This is what they're taking and this is what they're using. Now I'll let you read the rest, but I just wanted to read you some of that so you understand that where the thinking has come from and how you now can shift your understanding of causation. They have their lab-born lie of what a virus is, so they can give legitimate looking tests to unsuspecting hospital staff and workers that will then take stress products out of very diseased people who are diseased by the poisoning of their own government through the air quality. They will take that, they will compare it to their Western blot information and then say we have confirmed cases. You have patents for these particular blots and for the extractions of those phages in their stress forms where they make it in their own solution and make sure that they can get what they're looking for. Of course, they're patented. And why are they patented? So that they can push forward the vaccine afterward and make a crap load of money, all while maiming and killing and reducing the population, because they're all eugenicists and they don't like us and they don't want so many of us. So they want to make sure that they continue their, you know, kill, torture agenda, which you can find evidence of on the Georgia Guidestones, which you can find all over the internet. And yes, someone said that DuckDuckGo is bought by Google. And this is the other problem. Everything's going to keep getting bought up and bought up and bought up because they're writing their own laws. They're not, monopolies used to be illegal. Obviously, they're not now. They're going to just keep buying up the land. China is going to, I really think it's coming through China because China's got it really in the bag. They're going to push through the smart cities pretty fast and quick. They're going to push up the 5G as much as they can. So many people are going to die from 5G. It's really looking kind of apocalyptic right now. I don't know what else to say. I'm trying to educate people on what this is and how their little game, which really is linked in with everything. So I did a video on the climate change hoax, and that's part of it as well. And I also linked it with Hollywood and their players and how they're all in the big club and they have to play the agenda. And the agenda is UN Agenda 2030, have everybody in smart cities, have everybody microchipped, vaccinated, the perfect slave formed, people they don't like or the undesirables dead. When you're putting everyone in the mass concentration camp that they don't have as much stuff, right? So they want to encourage the tiny livings thing, and they use our desires to live small and easy and happy and well and have butterflies and happy birds and everything, and not kill animals and all the things that we truly desire. They are manipulating, they use that against us, right, to push in the environmental rules and regulations that they want to put in. He has perfected a new method of giving these DNA vaccines via electroporation. So what he does is he uses electricity to make pores in the cells, and then so it uptakes the DNA. That's frightening. They are going to try to genetically re-engineer us through these vaccines by giving us DNA vaccines, blasting us through our phones and our tech to make our cells porous to uptake their DNA. I hope that's making bells go off in your head. I bet China is the first real deep experiment of this. Turn on the 5G, give them the DNA vaccine. Let's see what happens. Are they all going to get cancer and die, or are we going to be able to do a mass genetically modified experiment on these people? That's what it looks like to me. I did genetic modification of cells in the laboratory, and it's exactly that. You throw the DNA in randomly and hope it gets to the area that you're looking for. And you put a marker next to it so that you can see if it has landed correctly. If things die, lots of stuff mutates, but then you get your one that you or two that you want. So I wonder if oh, if he hasn't done it already, Dr. Wiener, I bet he's already put in a marker to tell you when it worked in the human, because something will happen. And then they're like, we got the one. So what are they going to do with that person? Who knows? But this is a grand experiment on your lives. They are messing with your lives and your children's lives. Okay? When is it going to be enough for you to say, screw this? Okay, we're shutting this down. We're taking this to court, or we're exposing this in a huge way, or I'm getting my pitchfork and we're going to just do it ourselves.